swearing and saying controversial things now.
Sure. All right. Thanks everyone for joining us today. So it's my pleasure to introduce Paul. Paul is a professor in the Department of Mathematics at University of Utah. His research mainly focuses on stochastic processes in molecular and cell biology. So today he will tell us some of the related work about biological pattern formation. So Paul, feel free to start.
Okay, thanks. So Andy Oster was my graduate student. He told me that bars being the same time, so I'm amazed any of you are watching me. And but he's talking about neuroscience, and I don't do neuroscience anymore. So this is going to be about pattern formation. But I have to say, my interest in pattern formation goes back for many years when I did stuff on hallucination work with Jack Cowan. And I've always been interested in non-standard mechanisms for pattern formation because rather than the standard Turing-based mechanisms. So just let me remind you, let me just start straight away. There are three parts to this talk. I probably won't get through all of it, but what I want to mainly focus on is an area of interest of mine that hasn't been studied much from a mathematical viewpoint, but I think it's really interesting. Well, I wouldn't talk about it if it wasn't interesting. And so when I talk about pattern formation, I'm talking about two strands of pattern formation. We all know about Turing instabilities and the formation of spatially periodic patterns. But until really around 2000, there was very little evidence for the Turing mechanism playing a role in even animal coat patterns. It was only until Kondo discovered molecular mechanisms for stripe patterns in the angelfish, was it established that it actually does play a role as Turing had originally predicted. What dominated for many years was not Turing instabilities, but diffusion-based morphogen gradients. And so that's going to be my starting point in today's lecture. And just to remind most of you, I can assume you have a broad, at least basic background in most aspects of math biology, but please do ask me a question if something doesn't make sense. So if we look at the first slide, the standard picture of diffusion-based morphogen gradients, which goes back to a fellow Brit of mine, Lewis Wolpert, in the 60s, is that if you imagine there's some localized source of messenger RNA, and I like to be able to solve things, and I'm not very smart, so I tend to deal in 1D. So, well, everything's going to be 1D. No, that's not quite true. But just imagine a one-dimensional animal and along the body axis. And what happens is you have a localized source of messenger RNA that produces a protein, a morphogen, that then diffuses in the domain. And as it diffuses, it starts binding to receptors on the cells. And you end up in steady state. It's just a back of the envelope calculation that you get an exponentially decaying concentration gradient. And so a typical equation is, so I should say, when I first I recently gave this talk, and I didn't realize that when you switch the pen on, you can't change the slides, and I panicked. But so I'm going to risk it today. And so if you look at the standard diffusion equation, you have a diffusion term and a degradation term. But that degradation term really means binding to receptors in the cells. And as I said, you get this spatially decaying concentration gradient. And then that's converted to a cell differentiation, which is a discrete process where different gene circuits are switched on or off depending on the level of the concentration gradient in this thresholding. So you're converting an analog spatial code to a discrete cell differentiation code. And that's the basis of a lot of morphogenesis. And that's the standard picture.
So I have a patriotic remark, but we call that the French flag model, right?
Again, the last time I gave this talk, I said that, but as you're French, I ignored it. You're quite right. It is called the French flag model. So Jonathan's annoyed because I already mentioned Britain and its tradition in France to be annoyed by the British. So yes, quite right. It's the French flag model. Thank you. Do you know why it's called the French flag model? I have no idea.
Yeah, because if you had chosen white instead of orange in the middle, that would be red, white, blue, right, like the French flag.
So I'm explicitly anti-French here by not having white in the middle.
Yeah.
Okay. But what's been fascinating to me is in recent years, there's now growing experimental evidence that there's a different mechanism for morphogenesis based on cytonemes. So most cells extend these tiny thin filaments which are called philopodia or nanotubes, they're sometimes called, but I'm going to use the word cytoneme. It can extend up to about 200 microns. And the fascinating thing is, there are two fascinating things about these guys. First of all, they make direct contact with other cells. And second, it's been established that there's transport of either receptors or ligands associated with morphogenesis. And so the going hypothesis is that these play a major role in setting up morphogen gradients. And there's basically been, until recently, by myself and a very smart graduate student of mine, Hyung-Jung Kim, very little mathematical work on this. And so what I want to talk about is one of the versions of the model we've developed. And I just want to mention, just to be trendy, so if I was applying for a grant, I mentioned that there's been a lot of interest recently in terms of these philopodia within the context of the spread of COVID-19. So it's a really hot area. And that's something actually I'm beginning to look at with now, Hyung-Jung's now postdoc at UPenn, and we're beginning to look at some of these features. There's not going to talk about today, but what I wanted to do is talk about a particular model because it brings up a lot of the mathematical themes I'm thinking about these days from stochastic processes, but within the context of this particular application. And as I go through the talk, you'll notice there are quite weird things going on. And something that fascinates me, and I have no answer to, is how on earth cells develop this kind of hodgepodge of different mechanisms for solving problems. And it does really speak to the idea that in evolution, you take what's already available, you take it off the shelf, and then adapt it a bit to see if it works in a different context. So I always worry in a lot of theoretical biology these days, where it's sort of reverse engineering, where you think of cells or collection cells as these perfect engineered objects, when really, as far as I can tell, it's a sort of kludge. They take things that have evolved for different, solving different problems, and they adapt them to solve a new problem. So it's not optimized in many situations. That's not always the case, but certainly the sort of problems I've been looking at. Okay, so in invertebrates, the mechanism tends to be a cytoneme making a direct contact with a target cell, remaining fixed for a sufficient amount of time, and then vesicles of the morphogen or the receptor being transported inside the filament, motor-driven transport. There's still some debate about whether it's a direct contact like a nanotube or whether it's more like a synaptic contact. And we've modeled both cases, and there are differences. What I want to talk about today is a different mechanism, which I think leads to some really cool math. And it's a mechanism that seems to be observed in various vertebrates, in which the filaments are much more active. They're growing and shrinking. And it's been shown that at the tip of the cytonemes is a bunch of morphogen, or bunch of receptors. And what happens is they temporarily make contact with the target cell, deliver the cargo, and then retract. And so in this situation, the buildup of a morphogen gradient is based on so-called search and capture process, where these guys are growing and shrinking and eventually making contact with the cell, delivering cargo, and then retracting, reloading with cargo, and repeating the process. And there are many mathematical aspects of this which are really interesting. And there has certain analogs, for those who know about cell mitosis, of the search and capture model for microtubules looking for kinetic core connections on chromosomes prior to cell division. So there's been a bit of work on some of these things. So that's the basic setup. I'm going to spend most of my talk talking about the mathematics underlying our modeling work in this area. And so let's get going. And I'm going to try and go through some of the mathematical techniques. They might not be familiar to at least some of them. Okay. Now, one question that immediately I'm often asked, and I must admit I didn't have much of an answer until recently, is what benefits or potential advantage could it be for a system to use cytoneme-based morphogenesis rather than these cytonemes? Cytoneme-based morphogenesis rather than diffusion-based. And as far as I can tell, if you just look at 1D models, which we initially did, they can both do the same sort of thing. And in fact, diffusion you'd expect to be longer range because these cytonemes only extend typically up to a few hundred microns. But what I've realized by more recent work we've been doing is that if you go to high dimensions, you have much more control on the morphogen field you can create in 2 or 3 dimensions compared to diffusion. And so my suspicion is that it's in that context that morphogenesis based on cytonemes could be have a major advantage. Anyway, that's aside. So this is the basic picture in light of that. Let's imagine that we have, so now I'm going into applied math mode. I've given you the biology. I love doing math and applied math, and so I'm going to go into applied math mode, but there is this underlying biology in this stuff. So let's imagine a domain omega, 2 or 3 dimensional, with a distribution of target cells in it. And let's take each target cell to have a position at its center of xi. Let's imagine one source cell. Now, of course, I'm doing my modeling a spherical cow thing. I'm just taking one source cell with one cytoneme. But as long as you have independent growth of cytonemes, you can scale this up to n cytonemes. But let's just look at the statistics of a single cytoneme. And let pi be the probability that in one search and capture stage, it's growing in a direction in which it will if it manages to reach the ith cell. So there's already a probabilistic aspect to this, that each time it grows, it will nucleate in a different direction, just as you find in cell mitosis. And so there's a certain probability that when it's growing, it hits the ith cell. In that context, particularly in these 2 or 3 dimensional domains, there's also a non-zero probability that it grows in a completely useless direction and doesn't find any target cells. So we have to allow for that in that we sum the total probabilities of finding the kth target cell and take it to be less than 1. Now, in terms of the dynamics of the cytoneme, the second level of stochasticity is the following, that a cytoneme grows at a constant rate, but we allow, based on experimental evidence, that there's a certain probability that at an exponential rate, it can switch to a retraction phase. Now, and for those of you who know about some of these processes, this is a form of what's called stochastic resetting. And the reason one would think that this can happen, and again, it does happen in cell mitosis, is that if it happens to be growing in a useless direction, you don't want it to keep growing, it could be beneficial for it to randomly start shrinking again so it can search in a new direction. And this is an example of a stochastic search process. There's been a lot of work in many different applications where stochastic search is beneficial when there's a probability of failure. And that's in fact what we show. So that's the basic setup. So let's now put some mathematical meat on this. And so this is the basic schematic diagram. We have a source cell, and let's for the moment suppose that it's going towards a particular target cell. It's growing at a growth rate of V+, but at some exponentially distributed time, it can suddenly reset and go back to the source cell. And then after a waiting period, start growing again. Now in the full model, it then grows in a different direction. Here, if we're just thinking about a simplified model, we allow for the fact it could grow in a different direction by assigning a probability P that it grows to the given target cell and a probability 1 minus P that it grows in a non-direction, a direction that's not delivering to that cell. So in terms of the mathematical PDE, we have a hyperbolic PDE in which we take P plus to be the probability that the tip of the cytoneme, which contains a morphogen, is at a distance x from the source cell. And so we have a advection term representing growth. And then if it starts shrinking, we have an advection term representing shrinkage. And then we have a probability, as I said, an exponential distributed probability that at any time it can transfer from the growth phase to the shrinkage phase, which is given by these terms. Now, once it returns to the origin, the nucleation site, it stays there for a while. There's a refractory period, so there's refractoriness in this model, and that's given by the third equation. P naught's the probability that its zero length is in the nucleation phase, waiting a certain time, determined by eta, and after that, it starts growing again. So if you look at the terms here, this is the flux into the nucleation site here, and this is the rate at which it starts growing again.
So once the extension is retracting, it will always go all the way to.
Right. So we have developed models where it can switch back to the growth phase, but it simplifies the math. You don't get anything significantly different, but this is a simpler mathematical paradigm. You could, in fact, allow it to keep switching between growth and shrinkage.
So we can see it as, in fact, a reset of the direction after a delay that will
right.
Be the growth.
So the reset in the direction can only occur when it goes back to the nucleation site. Of course, cytonemes aren't completely rigid, so there's going to be some curvature, and we ignore that.
Right.
In this model.
But the retractation is completely deterministic, so it's like resetting the direction with a delay that is dependent on the length of the process.
Exactly.
Yeah. Okay.
Now, you can apply these resetting models to diffusion, so the whole process can be stochastic, and but generally speaking, the resetting mechanism is deterministic, but the time it occurs is stochastic. And so based on what I've said, then there are boundary conditions. The flux from the nucleation site back to the growing phase must balance the rate at which you leave the nucleation site. And if it does succeed in reaching the target cell, it's absorbed. And then we'll have initial conditions. We'll assume that time t equals 0 is at the nucleation site and it starts growing in the positive direction. And just to remind you that R is the resetting rate and eta is the nucleation rate. So that's the model. And it's a very simple model to write down, and it captures the essence of what's going on, given just one target cell. But even this is quite cool to analyze. And that's what I want to try and talk about. So I have to do a bit of basic stochastic process theory. Hopefully most of you have seen things like this before, but I'll try and go slowly. And I'm not that bothered about if I reach all the other topics. I think this is such a cool area and lots of cool math. Even if I just cover this topic, that would be fine. Okay, so given the boundary conditions, if we ask, so you can think of the integral of pxt dx, that's a survival probability. It's the probability that it's not in the nucleation state, nor has it been absorbed by the target. And the rate of change of that survival probability, or partial survival probability, plus the rate of change of the probability that I'm in the nucleation state is given by the flux into the target. So what's happening? There are three possibilities. I'm growing or shrinking, but I'm in the cytoneme. I'm at the nucleation state, or I've been absorbed. And you have to have conservation of probability, and this equation is the conservation of probability base. And you can get this mathematically by just using the evolution equations. Now let's introduce a survival probability that says I haven't been absorbed yet. So I'm either within the cytoneme growing and shrinking, or I'm at the nucleation site. The first passage time density to be absorbed by the target is then, this is a standard stuff in first passage time theory, is given by minus the derivative of the survival probability with respect to time, which from what I've done up above, the first equation is just the flux into the target. And so one of the quantities one might be interested in is, well, how long is it before it gets absorbed by the target on average? And that's the mean first passage time. And just by integrating by parts, basically T, the mean first passage time, is just the integral of the survival probability. And this is kind of, you see this in all different stochastic process contexts. Let me just remind you, ultimately, we're interested in establishing the existence of a morphogen gradient. This sort of mechanism is going to lead to a morphogen gradient in there are two aspects to it. When you have multiple targets, there's a so-called spreading probability. What's the probability I find this particular target rather than another? And then the question is, how long does it take me? Because now you imagine you repeat this many times, or you have many cytonemes, the buildup of the morphogen is going to depend on the time it takes to reach this particular target and how often it reaches it. So the statistics of the morphogen gradient, as I'll show later, depends on these two factors: the probability of finding the target and how long on average it takes to find the target. That's why these statistical quantities are important. Okay. So, but this is quite an unusual system, and I've come across these types of problems in many applications, including things like escape from domains where you have opening, closing ion channels and so forth, where, just to talk about that for a second, suppose you have a bounded domain, so it could be the cytoplasm, and you've got a molecule diffusing around, and it's going to escape through a hole in a pore in the membrane. This is often stochastically gated, so it's opening and closing randomly, and there's a quite strong possibility that when the protein reaches the pore, it's closed. So it's not going to escape, and it's going to carry on diffusing around. It's going to come back, and then hopefully this time it's open, but it might not be. In order to solve this problem, to estimate the probability that it escapes and how long it takes on average, you have to keep track of each time it hits the boundary and doesn't escape. So you have, if you like, you've got to keep track of, in some sense, there's a sort of memory and of previous events, and you have to keep track, you have to condition on these events. And an analogous thing happens here. So there's a great number of problems in stochastic processes where you have to condition on some random events. And I want to try and bring that out. If anything else from this talk, if you have never seen this before, to get a feel for this sort of method, I think it's a very useful method that's going to grow in importance. I'm saying that because I work in it, so hopefully it'll grow in importance. Okay. But it does exploit a form of memorylessness, and that's not a word, is it? It exploits the fact that there is a resetting which removes all memory of previous search phases. And for those of you who know about a lot about stochastic processes, because it's conditioned on an event that occurs at random times, one's exploiting what's called the strong Markov property. So the standard Markov property says that given my current state, I don't have to worry about prior states to determine what's happened at the next step. The strong Markov property says that still holds even if my current state occurs at a random time. So that's the basic thing we're going to exploit. So in order to do this, let's look at the figure, which is, say, one sample trajectory of this cytoneme. So what's happened is, so I've got this domain, and this represents the growing and resetting cytoneme. It might grow for a bit, and so it's just a straight line because it's constant growth. Then it resets and returns to the, I'm sorry, let me just get this, move this up a bit. Oh, that doesn't matter. Okay, so it then resets and shrinks at a rate v minus. Then it waits a random time tau before it starts growing again. And this can happen multiple times. But eventually, it will hit the target. In order to solve this problem, we break it up into a number of events. So let tau be the random time that it first is absorbed by the target. But let's also introduce some other stopping times, first fascist times. Let curly S represent the time that it first resets. So it might not reset at all, but this is saying, let's imagine that it does reset at least once, and the time for the first resetting plus growth is given by S. And then R, and then you have a randomly distributed refractory period, and then you have the remainder of the trajectory. The remainder of the trajectory could have 0 resettings, 1 resetting, 3 million resettings. So I'm breaking it up into conditioning on at least 1 resetting occurring and then the rest. Now it might seem a bit weird to do this, but this allows us to actually solve this problem in a very neat way, as I'll show in the next slide. Okay, so sorry, I mean, if you haven't seen this before, try and keep this picture in mind. So now let me introduce a few event spaces. Let omega be the set of all events which the particle is eventually absorbed by the target. If little p is 0, that has measure 1. So we're not allowing for it to grow in non-correct directions, so it just has measure 1. Let gamma be the subset of events for which there's at least 1 resetting. So that means the S term here is non-zero and it's less than capital T. All right. It immediately follows that if I take the omega slash gamma, that's the set of events for which the particle is captured without any resetting. So those are the different events, and it's almost like magic because it turns out you can apply this method to very complicated processes. I'm just trying to give you the simplest one. So what's the basic method? I'm interested in the expectation that the mean first passage time, which is the expectation of the total time t. But I can break that up into two parts. I can consider the contribution to the times, the trajectories that don't reset at all. And that's given by this guy. And then I have to consider all the trajectories that reset at least once, which is this guy. By doing that, I can then calculate these things in a very nice way. So that's the basic method that applies to lots of different things. So now let's calculate the various terms. So as I said, we have an exponential distribution of resetting times. So the probability of no resetting up to time t is just e to the minus rt. The survival probability without resetting is just a Heaviside function, because if I don't reset, I just have deterministic growth. As long as t is less than the distance between the target cell and the source cell, which is L, divided by the growth rate V plus, I still haven't been absorbed. And so then if I put all this together, I can calculate the expectation of the mean first passage time for all trajectories that don't reset. And basically, it's Laplace transforms occur all the time here. It's given by, with a bit of math, 1 plus r dr q naught tilde r, where r is a resetting rate. And because I have a very simple formula for my survival probability, I can explicitly calculate the Laplace transform of the survival probability. So I now have one term in my calculation of the mean first passage time. What about the other term? That's a bit more involved, but it comes, it exploits a really nice feature, as I'll try and explain. Okay, so let me just remind you about the this picture. If I reset at least once, I know that t is given by s plus tau plus r, the remainder of the trajectory. So if I plug that into my calculation, I then notice that the expectation of tau is just the mean nucleation time, which I'll call tau bar, which is 1 over eta. And then this is the crucial ingredient in these sorts of methods. It's the memory-less feature, the strong Markov property. Once I've got back to this point, because I've lost all memory of what's happened before, I'm back to basics. I'm back to the starting position. So the distribution, the statistics of R is identical to the statistics of T because of the losing the memory of previous state. And because this occurs at a random time, it's not the Markov property, it's the strong Markov property. So given that, I can take the mean of R to be the probability that there is at least one resetting times T, which is the thing on the left-hand side. So I'm basically getting an implicit equation for the total mean first passage time. And this lies at the heart of this. Even though you end up with quite a non-trivial result, it's exploiting this strong Markov property. Okay. The final ingredient is, so the two final things I have to do is calculate this guy and this guy. So let's calculate the expectation. So let me remind you, ES, given that I'm in this set of events gamma, is the expected time for me to grow and reset once. And I can calculate that, following on from Jonathan's comment. I mean, the reset process is deterministic. And so what I do is I say, well, I know the probability that I've reached a point X. I've been traveling at V plus, and then given that, I then take a time that which is given where I am, I take a certain time V plus, sorry, let me explain this a bit better. So V plus T, so let T be the time in which I reset, and that's distributed. V plus T is the current position I'm in. Then I reset at a rate V minus. So the total time to reset is V plus T over V minus. So that I know explicitly. I also know the probability of reset is given by this exponential distribution times the probability that I haven't been absorbed. So I put all that together and just do a bit of calculation, I end up with an explicit expression for this term, again involving the Laplace transform of the Q0, the survival probability without resetting. Almost done. So it looks a bit weird because this is quite an unusual way of thinking. And as I said, this is a simple example. But once you've done this a few times, it becomes sort of instinctive. I realized because when I first started doing this, I got very confused. So, but at least if I be aware of what's out there and you have the recording, I'm happy to send the talk as well if anyone's interested. Finally, I have to work out the probability there is one resetting event. And again, that is easy to do based on everything I've defined. Again, I use the strong Markov property, that the probability that R after one resetting is less than infinity is the same as probability T is less than infinity. And I've already said that has measure 1. And then using various calculations, one can also easily calculate the other guy. But as I'm kind of taking a bit of time on this, let me just go to the final result. So putting all this together, I get an implicit equation for T. On the right-hand side, I have the Laplace transform of the survival probability without resetting, evaluated at the resetting rate. And then I have a term involving the growth and shrinkage. And then the final term involves T itself, based on the strong Markov property. And this basic structure applies to many, many different models of stochastic resetting. Of course, the details are different, but basically they all end up involving an implicit equation for the first passage time that involves the survival probability Laplace transformed without resetting. And that's true for many, many different stochastic processes. And if you rearrange that, you end up with this beautiful result. And as I said, this applies to a wide range of different stochastic processes. This is the example. If you plug in the particular form for Q0 tilde for the CYTONEME model, you end up with this expression, which is very explicit. So that's the basic idea. And this, as I said, holds if your process without resetting is diffusion, advection diffusion, you name it, a velocity jump process is general velocity jump process. If you know what the behavior of the system is without resetting, you can then use these sorts of strong Markov property conditional expectations to determine the mean first passage time with resetting and express it in terms of statistics without resetting. This is a very powerful method that's been applied extensively in the past few years.
Can I ask a technical question, Paul?
Yeah.
So your process is piecewise deterministic. So you could know when we will hit the target. I mean, you could generate a sequence of exponential random variables, a discrete one, that would be the R, the time of switching. And what you really want is to have about, one of these exponentials to be larger than whatever the distance to the target divided by the speed.
Yeah.
And so you could generate like a sequence of such exponentials and take the first one that is larger than this value. And you could potentially get the same formula with that. I mean, how do the two methods compare? I'm sure you get the same expression.
You get the same result, and I've done that. So another way to say what you're doing, you can actually derive an integral formula, which is a occurrence relation. So you can do that in a different way. But I found, and so other people have developed that method along the lines you're talking about. But this, I find, once you get used to it, is much more efficient in deriving the results. And it's particularly effective when you have multiple targets and things like that. Yeah.
Thank you.
Okay, so I'm not going to redo it because you'll throw something virtually at me, but you can redo this for more complicated situations. I just want to point out that, so for example, I'll just show you schematically, if you include the situation in which you allow the system to fail to find the target, so you now take P to be less than 1, you can extend this analysis. And many other different things you can do. I'm just going to show you pictorially what's going to happen. So let's imagine with probability P, I start growing. as we've already discovered, so if you just look at the right-hand side here, so ignore everything else. So on the right-hand side, this is what I've talked about so far. With p equals 1, I start growing. I could reach the target without resetting. Alternatively, at some random time, I reset before finding the target and then repeat the process, exploring the strong Markov property. But if I allow p to be less than 1, each time in the nucleation sites, I could go to the left-hand side, in which I grow in the wrong direction, and then after some random time, which could have a different distribution to the nucleation time, for example, or resetting, I return to the origin, and then I shoot out in a different direction. And this lies at the heart of why having random resetting is so important for these sorts of processes, because if I have p less than 1, because there's a non-zero probability, there are a finite set, measured non-zero set of trajectories that don't lead to absorption by the target, the mean first passage time is infinite. Okay? You have to include those trajectories when you do the mean. However, if you allow for resetting, what you can prove is that with resetting, you can make the mean first passage time finite. That's the first point. But even more interesting is you can actually optimize the search process. By changing the resetting rate, there's an optimal value, and that's shown in this figure, where you get the minimum mean first passage time. And that's what's caused a lot of excitement more generally about stochastic resetting processes. There's an optimization going on. You can understand this intuitively, because as I said, if p is 0, you're infinite. But you're also infinite if the resetting rate's too large, because you start growing, and before you even have a chance to breathe, you reset to 0. You would keep resetting to 0 so quickly, you'll never find the target. So again, the mean first passage time is infinite. And in all the examples I've seen, and for many different applications, you tend to get a unimodal distribution and some optimal resetting rate. And that comes about by applying the methods I've talked about and then just plugging in numbers and plotting the curves. So it's a really interesting phenomenon, and also kind of explains why you do get this random behavior of cytonemes in the real system. And that's one of the things I like about, this is the sort of way I do mathematical biology. I like to do cool math. And I tend to deal with conceptual models rather than detailed data-driven models, because I like to see the connection between the mathematical aspects of this and why something might actually occur in the biology. And this is just one example. Okay. Multiple targets. So I could again get you to throw something at me virtually by repeating all this for multiple targets. I won't do it, but I'm just telling you, you can do it. And so now you have to say that there's a certain probability, pi j, that when during this process of going in different directions, I eventually find the jth target. And that's given by this formula. So you can push all this out. And you can also look at the conditional first passage times. Given that I find the jth target, what's the mean time to find that target? And you can calculate all of that explicitly. But you can read one of my papers if you're having difficulty sleeping, you suffer from insomnia, just read one of my papers and that will sort you out. But you can do it. So it looks like I'm just going to talk about this one project, but that's fine, rather than rushing it. I think there's so many cool mathematical aspects to this. Of course, I'm biased, but still. So I'm just going to focus on this today. So the next aspect, which brings in another very interesting aspect of stochastic processes, is the following. As I said, so far I've just focused on one cytoneme doing its thing and eventually delivering stuff to a target. Well, that's not going to be much use because the morphogen is going to degrade, and there's no notion of a morphogen gradient, which is ultimately what we're trying to do here. However, let's now consider the process where you get multiple search and capture events. And so this is shown schematically in this diagram. So you get what I've been talking about so far, growing, shrinking, going in different directions, finding a target. Now I add the extra thing that whenever the cytoneme delivers a target, you get a morphogen burst. And by the way, this is quite analogous mathematically to the transcriptional bursting in gene networks. Some of the mathematics is quite similar. Once it delivers its burst, it retracts, and we repeat the whole process. So now we take the stochastic search process I've been talking about for most of this talk, and now we repeat it. And we try and build up the distribution of morphogen across the whole population of target cells, given there's also degradation. Given degradation and delivery in this multiple search and capture process, one's going to reach a steady state distribution of morphogen across the population of target cells. That's our morphogen gradient. And so we have to take this complex stochastic search process for a single search and capture event and now put it into a model where there are multiple search and capture events. And that's the final element of this talk, because there's a really nice mapping between this process and queuing theory. And again, I'm not going to, I hope I'm not, no, I'm not going to go into all the details, but you can then get off the shelf methods in queuing theory and solve this problem. So I just want to give you the picture. So in queuing theory, you have a stochastic distribution of arriving customers that form a queue. Then there's one or more, it could be an infinite number of servers that at a certain rate serve the arriving customers, and then the customers exit. And this is a huge, there's huge literature on this in many, many different applications of queuing theory. How does it map onto this model? Well, the arriving customers are the morphogen bursts. So each time a morphogen burst arrives at a cell, that's an arriving customer. Instead of a server, we have degradation. And because the morphogen bursts can degrade independently, there are no constraints on when they degrade, that's equivalent mathematically to having an infinite number of servers. So this actual model is an example of a queue, and it's a particular form of queue called a GM infinity system. The point is that for wide range of stochastic processes, the statistics of the delivery of morphogen bursts tends to often be non-Markovian and certainly is quite general. The degradation process, the serving of the customers, is Markovian, that's the M. And because there are no constraints on degradation, we have effectively an infinite number of servers, and that's why it's called a GM infinity system. Okay. As I said, you don't have to know queuing theory just to get at least you can see the connection between arriving customers and morphogen bursts. So how does that relate to what I've been talking about for the first 3/4 of the talk? Well, in order to solve the queuing problem, I have to specify the G, the distribution of arriving customers, arriving morphogen bursts. Well, that relates to the first part of the talk, because the distribution of arriving customers is given by the splitting probability that I hit the Jth target, and then the cumulative distribution of the first passage, conditional first passage time for arrival of morphogen bursts. So these terms I calculate using all the methods I've talked about so far, and then I plug that into an off-the-shelf queuing theory formulation, which is really nice. I'm not going to go through the analysis. If you know queuing theory, it's pretty standard. If you don't know queuing theory, never seen it before, again, you'll throw something virtually at me. So instead, I'm just going to tell you the results. So M bar K is the steady state number of morphogen packets delivered to the kth cell. It is, as a function of K, the morphogen gradient. And it's given by, by the following. Gamma here is the degradation rate. Pi K is the splitting probability that I find the kth target. Tau kappa is another term I haven't really defined, but it says that when you're doing these multiple search and capture events, the dominant delay time is actually going to be the time to load up with new morphogen and the time to deliver that morphogen. It stays attached for 10 minutes in practice, delivers its morphogen, then retracts. And T is the unconditional mean first passage time. So one way to think of this is that 1 over T plus tau kappa is the frequency of morphogen deliveries, irrespective of the identity of the target cell. And then you have to weight that by the probability that the kth target cell picks up morphogen. And that's the result. The other really nice thing is you can use queuing theory to look at the statistics. and I've been exploring this quite a lot, where you can ask, well, what's the variance, the noise in the morphogen gradient? And that's important because morphogen gradients ultimately instruct cell differentiation. And, you know, the cliche stochastic genotype, regular, reliable phenotype, that's partly associated with robustness to noise. And that's something we're looking at. But I'm going to focus on just the morphogen gradient. Okay. Final thing about this. Let's take a simple example, because now the one thing we have to do, we've got all the ingredients, but the one thing we have to do is just to remind you that, sorry, go back a bit, the pi j's and other quantities depend on the probability that when I grow in a particular direction, I hit the jth target. That's going to be a geometrically determined quantity. And so I have to calculate pi j, and that will depend on the geometry of the cytonemes and the tissue that's being targeted. So let's again put on our applied mathematician hats and look at a simple example. I'll talk about 1D, but it extends to 2D and so forth. So let's imagine again, I have a single source cell, and there's a 1D array of target cells, and each target cell has a certain length. And each time the cytoneme grows, we'll take the direction of growth between 0 and pi over 2, which is uniformly distributed. Given those ingredients, I can just use a bit of trigonometry to actually estimate the probability that when I grow, I hit the kth target. Okay. So now I know pi k. So that's the final ingredient of all this. And if I plug that in, this is the final result, I get a morphogen gradient. Which of course I do, because if I didn't get a morphogen gradient, I just got some sort of fractal-like structure, this would be a particularly useless talk. So basically putting all that together, so let me just try and summarize why this I think is very interesting. Think of all the mathematical and modeling ingredients in this. We have a model of a single cytoneme growing and shrinking, with a stochastic component being the direction it grows in and the time in which it resets. Using these techniques I've talked about today, conditional expectations and the strong Markov property, I can determine the statistics of a single search and capture process. Splitting probabilities, if you have multiple targets, conditional mean first passage times. I can even calculate the conditional first passage time density. Then I want to port this into a process where you have multiple rounds of search and capture events to generate the morphogen gradient. I use queuing theory to do that, combined with geometrical trigonometric arguments to calculate these probabilities pk. When I put all that together, I can determine the mean number of morphogen across this one-dimensional target array and plot it in steady state and show how it behaves as a function of different parameters such as the resetting rate, and I get a morphogen gradient. And the reason why I think this is very powerful is that if you start thinking about two-dimensional layers or even three-dimensional structures, there's a lot of scope for really, if you like, carving out the morphogen gradient, which is not just a gradient now, in higher dimensions, which is much more difficult to achieve using diffusion. And just to also point out another important quantity that again reflects properties of stochastic resetting, if I ask, well, how does the total number of morphogen delivered across the tissue change with parameters? Just as the mean first passage time, there's an optimal rate of resetting in which I'll get the biggest bang for my buck. I'll still get the same weighting, the pi k's, but the total amount delivered is optimized at a particular resetting rate. And that's another result that comes out of this. Okay, so final point, I have simplified this by focusing on a single cytoneme from a single source cell. If we have multiple nucleation sites on a single source cell and or multiple source cells sending out cytonemes, as long as the nucleation processes are independent, I can just use the law of large numbers and talk about how these scale up with n. And as you all know, you're basically going to get a reduction in the noise, the variance thing that I calculated, going as 1 over n. And so this process, as long as there's independence of the different cytonemes scales up when in the more realistic situation where you, of course, have multiple nucleating cytonemes, etc. One final twist on this, though, is if you have, as you do in practice, multiple source cells, the other picture here, if you have multiple source cells, this guy, then of course the geometric construction becomes more complicated. This is still fine because they're all coming from the same almost point source, and so the geometric arguments extend. But of course, if you have extended source cells, then that aspect becomes more involved. And so in summary, combining these nice methods of conditional expectations and strong Markov properties, recurrence relations, stochastic resetting, queuing theory, and geometric constructions, one can now talk about the sorts of distribution resources across more realistic geometric distributions of target cells based on CYTONEME-based morphogenesis. And already a paper has come out applying these sorts of ideas to, it's computational, so I wasn't involved because I'm terrible at computers and things, I apologize. But applying this to more realistic scenarios and seeing some of the sort of things that occur here. And with that, I'm going to spend, I'm lying, the next 2 minutes going through the other half of my talk. But I won't. So I recently gave this talk to the UK, and I tried to get through all the material, and it was nuts. It was as if I was on cocaine or something, I don't know, or speed. And I thought this time I'll learn my lesson. I think this is a cool topic. So I thought I'd just focus on that. So I won't even talk about it, but I'll stop there. Okay. Any questions, comments, whatever? Oh, hands up. Oh, how do I do this? Ian. Did you have a hand up?
Yeah, I have a question, Paul.
Oh, all right, go on, Jonathan.
Was wondering, so yeah, it's very interesting. I was wondering, so compared to the classical kind of diffusion from a source theory, if I give you some kind of profile of concentration of morphogen, you can predict from this theory, and I give you so the distribution and the architecture, I mean the geometry of the problem. You can compute probably the statistics of the search process. Do you think you can do such predictions? And I mean, what are kind of the necessary conditions you have on the profile of the, what kind of class of function can you generate?
Right, that's a good question. So, so the answer is I don't know, but a few things to point out. So, and this is true for diffusion-based, that one thing one could do is look at the length constant of the gradient, which is something. There's also something else. You'll notice this behavior over here is very different from the standard exponential decrease. So one prediction would be, if this is the mechanism, you know, because people also play around with algebraic decay. So if you have nonlinear degradation in a diffusion-based model, you get this monotonically decreasing function, but it's given by algebraic decay. But none of them seem to show this behavior. So that would be one marker that maybe there's a cycling-based process going on rather than the standard diffusion-based. And more importantly, it's something I haven't worked out yet, I think going to two-dimensional and three-dimensional target tissue, the sorts of distributions one can get should be much more varied than the standard things you get in diffusion-based, which are going to be expanding rate, you know, spherical distributions and things like that. This could be much more complicated, and I think it lies at the heart of why this might actually occur, at least in some morphogenesis. But that'd be an interesting problem to push that further. Yeah. Any other questions?
Yeah, thank you for the talk. I'm just curious, do you have any pictures or videos of what maybe the steady states of these things look like?
The pictures of, you mean from the biology?
No, I mean maybe simulations or...
Oh, you said the word
just
simulation. You don't know.
Or, you know, it could be the result of theory. I don't know. Just something to show what, I don't know, is hiding behind these probabilities. I don't know.
Oh, no.
Does it look different than diffusion?
But I mean, this is, so the only two are these that I've included. So this is an example of the results of all the processes. This is the steady state distribution of the morphogen gradient, which inside it has the splitting probabilities and the total time t. So they're built in there. And I'm trying to think in my papers if I had plots of those. There are various plots of these, but those are the only things I include in this talk. But yeah, and also the total. So whoops, what happened there? Oh yeah. So you'll notice that MK bar is given by this. So built in there are the splitting probabilities. So if you like to answer your question, the plot of M bar K as a function of K is the distribution of splitting probabilities. So that's one statistic. And if you sum overall K, the sum over pi K is 1 if you assume there's no probability of failure. So M tot is the inverse essentially of the total mean first passage time. So two of the statistics are actually shown in these figures. So that's the inverse, if you like, of the total mean first passage time. And this is the distribution of splitting probabilities. Is that?
Okay, no, that answers it.
Yeah.
I one quick follow-up question is, at some point you mentioned something about bursts of transcription, or I just never heard anything like that before. What exactly is this burst of gene expression?
So,
that's relevant to this topic.
Bursting, that because you have low numbers of messenger RNA produced by a gene, and the messenger RNAs have a short lifetime, each time you produce a messenger RNA, you produce a burst of protein. And that's one example of protein bursting. And so you get very bursty behavior. So, so rather than seeing protein produced at some sort of uniform or approximately continuous rate, you get bursts of protein being produced. And that occurs due to stochasticity in transcription because of stochasticity of promoters binding and unbinding to operator sites. That's one source of stochasticity, and the other is just the low copy number. So both of these produce, rather than a sort of analog distribution of production of protein, you get bursts, analogous to the morphogen bursts. And if you're interested in this, if you email me, I can send you a reference to a paper that applies this queuing theory to transcriptional bursting. It's very, very nice. So there are a lot of parallels, which is the cool thing about math. You can apply these things to lots of different applications. And of course you get.
No, that's very interesting. I just never heard of it before. I'm somewhat of a biological.
So yeah, so and of course you get neural bursting, but they tend to be of a different form, and that's putting on my old hat.
Mm-hmm. Well, thank you very much for the talk. It's very interesting.
Yeah. And
I have a follow-up question if nobody else has.
Okay, go Jonathan and then Andy, I think, had a question.
Thank you.
Jonathan?
Okay, so my question comes back to one of the questions I asked in the beginning related to the choice of an exponential. Just the choice of looking at your process as a continuous time versus just a discrete Markov chain. I can see that the assumption of having an exponential search time is important in the continuous view of the process. And I was wondering if you had a search process that lasted not an exponential time, but had was centered around
So
time or something.
Yeah.
You could.
So that's looked at. So that is actually looked at. So I kept it very simple, but there's been a lot of work having non-exponential waiting times for resetting. You could also have non-exponential waiting times for the nucleation as well. So that has been done. And again, you can handle it because ultimately you go into a sort of Laplace transform space. So you can incorporate that. You're going to have more complicated, obviously, Laplace transforms. But as long as so I'm trying to think, I suppose in order for this to work, you need to assume that the moments of the non-exponential waiting time density are finite, at least the mean, to get this to work in any practical sense. But yes, certainly this machinery can be extended to incorporate non-exponential waiting times, for example.
But wouldn't you lose the
Markov property?
You would lose the Markov property for the continuous process,
yes,
but not for the discrete.
Yeah, so this particular method couldn't be applied. You'd have to kind of develop a different approach to recurrence and things like that. Yeah, that's true. Andy, do you have a question?
Oh, I was just was going to follow up on what I think it was Ian's comment. So it's not so much a burst as in the neuroscience, but rather more like a single spike. When you say burst, it's more like they're just spikes. So it's like a sequence of action potential, so to speak.
Yeah, it's not a slow, fast, manifold dynamical system. It's not an Izakevich burst or whatever. No, but
and then,
but it's, again, I have to say though, but it is burst in the sense that because of the dynamical systems process, you get packets of
clusters,
bytes. So in that sense, it is a burst. So like this is a packet of morphogen.
And then as far as this approach, you plan on going to looking at, you think you said Philopadia or Philopedia, I'm not sure the answer. Are you looking at certain sort of context? You're talking about the COVID, but I know this gets used something along this lines for growth cone dynamics for setting up just the visual system, for instance.
Right. So in the context, so I haven't looked at it in terms of neuroscience, but in the context of COVID, there it's very interesting because you have, so if you try and port this into an SIR model or something like that, you have to think about the timescales and dynamics of the stochastic process of growing a filament and finding a susceptible cell. And the timescales have to be related to the internal dynamics of an effective cell, infective cell, and so forth. So that's something we're trying to look at the moment. How do you combine this stochastic dynamics with the dynamics of SIR models and things like that? And it's non-trivial. So whether we'll make any progress is another matter, but that's the sort of thing we're doing, yeah.
Would it be SIR models or just like the nature of the immune system itself?
With the SIR models, that's what our initial step is, just to do that. But yeah, so this is within host spread of the disease, not across a population. Yeah. And I should say there's been it's not my field, so I'm just learning about this now, but there's been always a lot of interesting differences. There's, again, within host spread of infections, there's diffusion-based mechanisms where viruses spread diffusively, and then direct contact mechanisms. And people have been trying to look at the differences between the two within the context of immunology. So I think this is a kind of a very interesting general aspect to various applications of cell biology, difference between direct contact and diffusive mechanism for spreading stuff. And it also occurs in cancer. But I'm a hypochondriac, so I tend not to work on things that involve disease. So I'm not sure I'm going to pursue that too much. I worry about enough anyway, so. Yeah. Okay. Any other questions? I'm chairing my own talk. I'm not supposed to chair my own talk.
You're too active. I don't even have a chance to check in. Well, do we have any other questions for Paul? Well, it's been a very nice talk for today, and I think we'll just wrap up here. I'd like to thank our speaker, Paul, again for giving such an interesting talk and everyone for participating. So I guess we will have another talk next week, and I will see you all next week then.
Okay, well, thanks for listening, especially as Bard was talking at the same time, I think, or overlapping. And yeah, I enjoyed it. And this is a very rare event, because for me to just get through 24, 25 slides in a talk is unheard of. Usually up to 40 or something like that. But for once, I decided to kind of go a bit more slowly, because these techniques might not be familiar to anyway. But thanks for listening, and keep safe and well. Cheers, Jonathan. Cheers, Andy.
Bye-bye.
I'll see you guys
Thanks,
at some
Paul.
point. Bye.
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 Thanks, Paul.
Thanks for inviting me. See ya.